Hello viewers, welcome to my channel. Today we will discuss about the bacterial growth curve. Bacteria are prokaryotic unicellular microorganisms. Bacterial growth means increase in the cell numbers rather than increase in size or height. Bacteria primarily reproduce by binary division, in which one cell divides into two cells divide into four cells divide into 8, and so on. The time required the generation to double is known as doubling time or generation time. In my previous video, I have already discussed some mathematical problem on generation time on doubling time. Here the figure of binary division is shown. Now how we obtain the bacterial growth curve? So to obtain a bacterial growth curve, first we have to inoculate a bacterial culture into a suitable liquid culture medium. Then this culture medium should be incubated for a particular constant temperature and proper environment. The growth follows a definite growth pattern. The growth measured at regular time intervals and the values obtained will be plotted against a logarithmic graph paper. Show a typical growth curve. Here a typical growth curve is shown here, where time is in the x-axis and in y-axis log number of cells. Bacterial growth phase divided into 4 phases. The first phase is lag phase. Second one is log phase or exponential phase. Third is stationary phase. And the fourth one is declining or death phase. Now we will discuss each phase elaborately. First is the lag phase. After a liquid culture broth is inoculated, the multiplication of bacteria does not start immediately. It takes some time to multiply. The time between inoculation and the beginning of the multiplication is known as the lag phase. In this phase, the inoculated bacteria become acclimatized to the environments, switch on various enzymes, and adjust to the new fresh nutrient medium, temperature, and pH of the media. During this phase, there is an increase in size of bacteria, but no appreciable increase in the number of bacterial cells. The cells are very active metabolically. As we know that in case of bacterial growth, it means the increase in the cell number. So here the growth does not occur. Only the size increases maximum. During this phase, cell synthesize amino acid, protein, RNA, enzymes, which are further necessary for growth and cell division. So this phase is known as preparatory phase. As during this lag phase, cells synthesize their essential metabolites, nutrients, amino acids for their further growth and cell division. The duration of lag phase varies with bacterial species, nature of the culture medium, incubation temperature, etc. It may vary from 1 hour to several days. At the end of the lag phase, the bacterial cells start to multiply and the cells enter in a logarithmic phase of growth. The next one is log phase or exponential phase. This phase is very important. This phase is also called exponential phase. In this phase, characterized by exponential cell growth, that is from 1 to 2, 2 to 4, 4 to 8, and so on. The bacterial populations doubles during every generation. They multiply at their maximum rate. The bacterial cells are small and uniformly stained. So, in our lag phase, we have found that the bacterial cells become maximum size. Whereas in log phase, bacterium cells become small and uniformly stained. The microbes are sensitive to adverse conditions such as antibiotic and other antimicrobial agents during this phase. The growth rate is the greatest during this log phase. The log phase is always brief, unless the rapidly divided culture is maintained by constant addition of nutrients and frequent removal of waste product. When we plotted it in a logarithmic graph paper, The log phase appeared as a steeply sloped straight line. Next, the stationary phase. After the log phase, the bacterial growth almost stops completely due to lack of essential nutrient, lack of water, oxygen, change in pH of the medium, etc. Also due to accumulation of their own toxic metabolite. It is during this phase where the culture is at greatest population density. During this phase, growth rate is equal to death rate and the population stabilizes. The graph shows a horizontal line. This phase is also important as during this stage, endospores start forming. The bacteria become gram variable and show irregular staining. Many bacteria also start producing exotoxin. The last phase is death phase or decline phase. During this phase, the bacterial population declines due to death of cells. Here, death rate is high. The decline phase starts due to accumulation of toxic product and autolytic enzymes and exertion of nutrients. Involution forms are common in this stage. Some cells assume various shapes becoming long, filamentous rod or branching or globular forms that are difficult to identify. Some develop without a cell wall and are referred to as protoplast, severoplast or L-phase variant. When these involuted forms are inoculated into a fresh medium, they usually revert to the original shape of the healthy bacteria. So in brief, lag phase is the preparatory phase where the bacteria cell prepare for amino acids, enzymes for their further cell division and growth. Log phase where the growth rate is high and during this phase the cells are growing exponentially. The third phase is stationary phase where the growth rate is equal to death rate. The population stabilizes and during the stationary phase endosperm also form. And the final is last phase is the death phase, declining phase where death rate exceeds as due to accumulation of toxic product and exertion of nutrient. Thank you. Please like, share and subscribe to this channel.